Hi, everybody. This is the first lecture for week 4. We're going to be talking about infection and what this looks like from the virus's perspective. So I'm going to go ahead and share my screen. We'll head to my PowerPoint. In this one, we're going to look at the 5 steps of infection and thinking about it as though we are the virus. So this is kind of the fun time of where you get to pretend you're a virus and think about what it looks like of you trying to get in, cause an infection, and then get out. I will want you to be able to describe various portals of entry and exit for viruses. Most viruses have 1 or 2 particular places where they have the ability to enter and exit. We'll go through some examples of that. I'd also like you to think about the different mechanisms that pathogens can use to avoid immune detection, meaning that our immune system wouldn't be able to detect them and eliminate them. So many viruses have mechanisms that allow them either to hide or to make it so that the immune system is not as efficient. Then we have talking about how symptoms from different infections can actually help in that transmission of the virus. So I'm going to walk us through the 5 steps of infection. First, we'll do an outline of it, and then I'll go into more detail for each of these. So if I'm the virus, there's 5 things I want to do. The first thing I need to do is I need to enter the host. So I need to find a portal of entrance. I like to think about 2 different types of viruses that we might get an infection from. So one of them would be an upper respiratory infection, and the other one that I like to follow it through would be something in the digestive tract. So in the portal of entry, we can think about with respiratory, it would often be our sinuses up through our nose or in our mouth. With a gastrointestinal virus, we think of it as something we ingest through our mouth. The next thing that I want to do is if I get into the nose, right, or if I get into the oral cavity, I want to adhere or stick to the tissues. I want to do this because I don't want to be washed away with saliva or blown out of a nose. I need to figure out some way to attach myself. Once I've attached, I go on to what is called invasion, and that means that I enter into the cells of the host, sometimes even to underlying tissue, and then I can go through evasion and replication. In the evasion and replication, I look to make sure that the immune defenses of that host don't kill me off or destroy me. And if I can do that, it gives me the opportunity to start to replicate and make more virus particles. Lastly, when I've replicated and make many, many virions, I look for a portal of exit in order to be transmitted to a new host. So I'm going to walk through each of these and talk about them. The first, as I said, is the portal of entry, and this is any site that that virus could use to enter the host. Most commonly, we see this at mucous membranes, so in the nose, in the mouth, around the eyes, the genital and urinary tract area. This is often where the site of the disease would develop, but not necessarily the only place or where the main site would be affected. Often it can move to other parts. For example, SARS-CoV-2 might come in through the nose, but we know that sometimes it can have real devastation in the lungs, so in that lower respiratory tract. Some pathogens have more than one portal of entry. I have some examples of these, of preferred routes of entry. I will be talking about some different types of viruses, but please note I'm not expecting you to memorize them all. It's just to give you some examples. So viruses can enter hosts through skin or mucous membranes. I said the respiratory tract, that's one that we often get that can be aerosolized. We have the gastrointestinal tract. Often this can be something that is a fecal contamination that then somehow contaminates something and gets eaten. We have the genital tract that would be spread through sexual intercourse or sexual connection. We also can have conjunctiva, which is the area around the eyes, the mucous membrane then. And then we can also have the placenta. So in pregnancy, we can have some viruses that can actually cross from the mother to the developing embryo, and these are considered blood-borne viruses. There are some that are zoonosis or zoonotic, meaning that they are transmitted from an animal to a human. So for example, we may have some that are transmitted by insects that we get from insect bites. maybe a bite from an animal in the case of rabies. And then sometimes we can have where it has an insect that is kind of the secondary host that can also infect us. With the respiratory tract, this tends to be the most common place that we see infection from. These small particles can be inhaled through the nose. In our case, we're thinking about viruses. And because our body has this mucus that is in our nose and our upper respiratory tract, often what happens is that can get things to stick there. And we have cilia that will brush things up to try to get it to exit that cavity. We often cough and sneeze as well, that hopefully propels that out. So some that we can think of, though, that would infect us through the respiratory tract would be RSV, or respiratory syncytial virus. That's one that we know is very common in the winter. Rhinovirus, which is the cause of the common cold, and any of the coronaviruses, including SARS-CoV-2, would be ones we'd see through the respiratory tract. That's how, if I'm that virus, I'm going to enter a host. Gastrointestinal tract entry obviously is viruses that enter through the oral route. Usually that means then they pass through the fecal route, which if we don't properly use proper hygiene, we can then recontaminate things and pass it on to others. I have a list of them here. Don't worry too much about memorizing all of these, but this is to give you some idea. Rotavirus we commonly see in young children. Norovirus, the reason why most microbiologists will not get on a cruise ship. And then poliovirus is one, too, that we'll talk about a little bit later, that we can get through this as well, and some of the hepatitis and enteroviruses. But thinking about some of those stomach viruses, these are ones that might be the culprit. Sexually transmitted diseases are generally passed between the genital tract during sexual intercourse. We have ones that are probably familiar to, so HIV 1 and 2, 2 of the hepatitis viruses, herpes simplex and papillomavirus. We also know that Ebola and Zika can pass this way as well. Again, I'm not expecting you to memorize all these or the symptoms and disease, but rather to give you some examples of these different types of portals of entry as we, the virus, try to get into a body. The last few are ones that are slightly less common, but also can happen. We can have skin entry. So on the outer layer of the skin, we have dead cells. Usually that protects us. But if that virus can get in where we have cuts or breaks in the skin, that's often where they'll enter. The conjunctiva, right, the mucous membranes of the eye, this is a place where viruses can enter. Also in bone marrow or organ transplants, so in transplants of either of those two things, if there was virus in the transplant, that could affect the recipient of those. And lastly, we can get them through transfusion and blood products. However, we try to do a lot of screening, so that's not as common as maybe it used to be. So we got in our portal of entry. If I'm SARS-CoV-2, I came in through the respiratory tract, and let's say I went on a cruise, which I would not. Maybe I have norovirus, would be the portal of entry, would be through the intestinal tract. The next thing that we have to do is we want to adhere, right? We want to stick to something. So once we enter that host, we are usually going to do an initial adhesion that's non-specific, meaning that it's often through hydrophobic interactions that we kind of stick together. Once we do that, often we use then our spike proteins if we're a virus, right? We have those spike proteins that hopefully are perfect fit for the receptors on the cells at the portal of entry. And that's generally how viruses get in. They make that attachment. It's like a key inserted in a lock, and that cell then brings it in. That brings us to our third step. And that's when that pathogen is going to invade tissues. And so in this case, what happens is that spike unlocks that receptor, and the virus is allowed then to invade the cell. As it invades, it can often cause damage to the tissue or the cells. And when we see that with viruses, typically the damage that they can do to a cell is, first of all, they'll disrupt the normal host cell function because they'll be taking over the ribosomes, the enzymes, hijacking that whole cell to make more virus particles. They have to be released from the host cell, which in the case of naked viruses can destroy the host cell. And then some of them have the ability to actually transform normal cells to cancer cells. And this would happen during this kind of invasion period. Once we've invaded, you want to think about what's going on in the host cell. If I'm a virus and I'm starting to cause damage, we should expect that the immune system is going to be alerted to that, and it will likely start trying to come after us. So that's where our fourth step comes in, and that's that we have to be able to evade or hide out from the host immune defense so that we have time to replicate. There's three different kind of big ways that viruses have the ability to do this. We haven't gotten into the immune system yet, so I'm just going to do an overview of it. When we get into the immune system in the next chapter, I'll give a little bit more explanation. So the first way that viruses, or us, as we are the pathogen right now, can evade the immune system is that we get into a host cell, and often it's hard for our immune system to be able to see us when we're inside that host cell. Secondly, we might do something that's called immune system evasion. And this is something where we can actually affect the way that the immune system reacts. We can either inactivate some of the signals that our immune system uses to communicate. We might block some of the cellular pathways of the immune system. And also, if we're really good at mutating fast, it means that the immune system might not be able to recognize us, even if it was maybe it already recognized an earlier strain of us. If we mutate, it may not recognize us anymore. The third kind of big way that we can evade the host immunity is go into what we call latency. So in the week 3, I talked a little bit about that lysogenic bacteriophage. Animal viruses, some of them have the ability to do something similar to that and go into latency. So for example, HIV and the herpes family of viruses have this ability to basically quietly exist inside of a host cell by integrating their DNA into our chromosome, and they'll hide out there for periods of time. And often our immune system can't find them, and even when we try to use drugs to treat them, if they're in latency, it can't actually work. So if I went to the portal of entry, I attached myself, I invaded and got into the cells, and I managed to not get deactivated by the immune system, I can now replicate, and I will make millions upon millions of virions and be ready to release them from the cell. And that brings us to our fifth step. Our fifth step then is that pathogen now wants to be transmitted to a new host. So maybe I'm sick of being in this particular body, and now I'm going to be transmitted to another. So the last step in a pathogen success is that transmission to a new host. And any route that pathogen exits one body to get to a new host is referred to as the portal of exit. And you can think about many of the things that come out of our body as possible places where viruses could exit. So in our waste, feces, urine, any bodily fluids such as blood or maybe fluids that drain from a wound, even vomit and saliva or semen are all places where viruses can leave the body. Often the portal of exit is the same as the portal of entry, but remember that's not always true because of its norovirus. It may be coming at the other end of my digestive tract. One of the things to keep in mind is that often the symptoms that pathogen causes in the host will aid it in being transmitted from that host to a new host. So let's say you have some sort of a skin infection, or you have something in the respiratory infection. With skin infection, it might become itchy. You'll start scratching it. That will slough off some of the cells and also some of the viruses that could infect someone else. With sneezing and coughing, if we have lots of viruses in the snot or in our saliva and we sneeze and cough, that's a great way for that pathogen to go from one host to another host. And then diarrhea tends to be kind of messy. That means that there's likely a lot of virus that's shed in that diarrhea. And if we don't properly clean it up and use proper hygiene, it could contaminate something else and infect another host. So that kind of takes us through, as the pathogen, those 5 steps of infection, right? I go in my portal of entrance, I attach myself, I then do my invasion. I want to evade or escape the immune system and replicate, and then I'm ready to head out and find a new host. I will let you pause it here to do a quick checking in. and then I will come back and answer the questions for you. So the first question is, what is the first step for viral infection? Portal of entry is the first step. What structure aids in the second step? The second step is attachment. So the structure in viruses that would aid is the spike proteins. After invasion, what must a virus do to be able to replicate? It needs to evade or escape the immune system or immune defenses of the host, and then it can go on to replication. Are portals of exit always the same as portals of entry? They are not always, but often they are the same portals. If you have any questions, please let me know.